It's my honor to introduce our first speaker today, Dr. David Standaert. David graduated from Harvard College, another Harvard alum, received MD, PhD from Washington University in St. Louis, and following neurology residency at UPenn, and he was appointed as a Harvard Hughes Fellow and completed 3 years of research and clinical fellowship in movement disorders at the Massachusetts General Hospital. He was a member of the faculty at Harvard Med School from 1995 to 2006, and then relocated to University of Alabama at Birmingham. Currently, he is the John Whitaker Professor and Chair of the Department of Neurology, and a senior member of the Faculty Division of Movement Disorder. He directs the NIH-funded Alabama Morris Udall Center of Excellence in Parkinson's Disease Research. He's the chairman of Scientific Advisory Board of American Parkinsons Disease Association, a deputy editor of the journal Movement Disorders, and a fellow of both American Neurological Association and American Academy of Neurology. First vice president of the Association of the University Professors of Neurology, and a member of NIH and NDS Board of Scientific Counselors. His lab has a long-standing interest in basic mechanisms underlying Parkinson's disease, as well as the complication of therapy. Now, David, thank you for your lecture in advance.
All right, well, I'm happy to join you all virtually, and I hope I can come do it in person some year. But this is a great opportunity to talk with all of you about some of the things we've been working on here at UAB. Let me go ahead and get the screen sharing to work. Let's see. How's that? Is that working?
Perfect.
Yes.
All right. So we're going to talk about innate and adaptive immunity in Parkinson's disease. And to me, this is it's really remarkable to be able to speak about this. 10 years ago, I think if I had said I wanted to give a lecture on the role of the immune system in Parkinson's disease, very few people would have listened. It wasn't a very popular topic, and it's changed tremendously. There's a lot of interest in the field in this area. I think it's really turned around in the last few years and now seen as a major approach to both understanding the disease and new therapy. And I want to give you some sense of the excitement around this. In terms of disclosures, I do consult for a number of companies, but none of these are working in this space, and we're not going to talk about any off-label use of medications. I think it's always best to begin at the beginning. So this is the faceplate from James Parkinson's essay on the shaking palsy. This was his first description, at least in the Western literature, of Parkinson's disease. He published this in 1817. And he described it as an involuntary tremulous motion with lessened muscular power in parts not in action, and even when supported, with a propensity to bend the trunk forward and to pass from a walking to a running pace. Still a good description of the core features that we look for in Parkinson's disease and diagnosing this. And on the right here, some famous figures who have been associated and come out publicly as either advocates or affected by Parkinson's disease. Of course, the Pope John Paul, that's Davis Finney on the top right, winner of several stages of the Tour de France. Linda Ronstadt, that's Michael J. Fox there with Roger Daltrey, Muhammad Ali, Mo Udall, and of course, Robin Williams. And so many others have represented the public face of Parkinson's disease, I think, to the world. Parkinson's, the main risk factor for Parkinson's disease is age. And the good news is, of course, we are living longer in this country. At this point, most Americans who are currently alive can expect to live to at least 80 years. But the not so good news about that is the older you get, the more likely you are to get Parkinson's disease. And you can see from this graph here how steeply this rises. At early ages, before the age of 40, it's quite a rare condition. But as you turn the corner here from 50 to 59 to 60 to 69, and then ultimately into the 70s, it rises very steeply. And it's also more common in males compared to females. And this is something that persists across all ages. So as our population in this country and many countries around the world ages, we're going to see more Parkinson's disease simply because of the growth of the number of individuals who are over 65, which is going on throughout the US and actually throughout the world. So this is going to be a major force for the next 10 or 20 years as the so-called baby boomers age and the incidence or the prevalence of Parkinson's climbs because of that. Features of Parkinson's. This slide many years ago was titled Features of Parkinson's Disease. Now it's called Classical Features. These are the really basic things about Parkinson's that we teach even undergraduate courses or medical school. So clinically, Parkinson's disease, we look for a triad or a tetrad of symptoms. Resting tremor, a very particular tremor that's present when the limb is at rest and tends to go away when it's used. Bradykinesia, slowness of movement, rigidity, stiffness, resistance to passive movement, and then postural imbalance, a tendency to fall. And so these are the clinical features that we look for in Parkinson's disease to make a clinical diagnosis. And in terms of pathology, the well-known pathology of Parkinson's, this is a normal human substantia nigra here. This is the midbrain. The black substance here is the substantia nigra. It's black because of production of dopamine leads to neuromelanin, which accumulates in these neurons. In Parkinson's disease, there's very complete destruction of this nucleus. Typically at autopsy, 95 to 99% of the dopaminergic neurons are gone. There's certainly pathology elsewhere in the brain, but the destruction of the dopaminergic system is very complete. Microscopically, the classic feature, this pink picture in the middle, shows a neuron with a brown substance in it. That's neuromelanin. That's what it looks like under high magnification. And then this round sphere is a Lewy body. These were described really in the early 1900s. And for a long time, it was unknown what the composition of the Lewy body was. It was understood they were a signature of Parkinson's disease. They have an eosinophilic core and a clear halo. As we'll come to in a minute, we now understand the main component of the Lewy body is a protein called alpha-synuclein. There are a number of other proteins in there, but they are the pathologic signature of Parkinson's. And then lastly, there are now techniques to monitor the loss of the dopaminergic system in life. This is a set of images at the bottom here from one of my patients that was scanned using a technique that labels the presynaptic dopamine terminals. And he was scanned in a research study every year for 4 years. And you can see the fading out of the presynaptic dopaminergic terminals here. This is very similar to something called a DAT scan, which is used clinically these days. Very similar kind of tracer, binds to the presynaptic dopamine transporter, and is available clinically to actually study this in patients and used for diagnostic purposes. But the other thing we've learned about Parkinson's disease, there are a lot of other things going on, the so-called non-motor features of Parkinson's. And I really carved these into 2 groups. There is the so-called early or premotor features. These are features that are seen in Parkinson's disease often before the tremor, before the stiffness and slowness. Include things like hyposmia, the loss of sense of smell, which can come on 15, 20 years before the other symptoms of Parkinson's. REM behavior disorder, this is rapid eye movement sleep disorder. Patients who act out their dreams, thrash at night. Turns out that this was originally believed to be an unrelated condition, but now is recognized really as a precursor of Parkinson's. And the longer we study it, the more we realize that pretty much everybody with REM behavior disorder goes on to develop Parkinson's disease. Most studies suggest it's between 50 and 80% of people with REM behavior disorder will eventually develop Parkinson's. And autonomic disturbances are increasingly understood as an early feature of Parkinson's. Cardiac denervation, blood pressure discontrol, other kinds of autonomic aspects. There's also a group of late features: excessive sleepiness, depression, anxiety, and then ultimately dementia. Dementia is not a universal feature in Parkinson's, but in patients far into the course, the incidence may approach 50% or more. So it does become a major problem late in the game. So with all of this, what's the treatment of Parkinson's disease? Well, I think, as I said, we started with Dr. Parkinson. Here's Dr. Parkinson's treatment. He suggested bloodletting, which in 1817, this was a treatment for everything, very common treatment among physicians at that time. And he recommended taking blood from the upper part of the neck. And that was the treatment of the day in 1817. Interestingly, he actually suggested creating an inflammatory state. So had some thinking that inflammation might have a role in Parkinson's even more than 200 years ago here. Today, in 2020, we have no treatment which can slow or prevent the progression of Parkinson's disease. There are a lot of symptomatic drugs. I spend a lot of time in clinic treating patients, and I think we do a great deal of good. We help a lot of people with symptomatic treatments, but at the end of the day, there are no treatments which have been established as preventing or slowing the progression of Parkinson's. And this is where the focus of research really needs to be. A few words about the genetics of Parkinson's. Now, this is another area that's changed a lot. If you read a textbook from 15, 20 years ago, it will say Parkinson's is not a genetic disease. And in fact, in most patients, it's not unusual to have them say, oh, I had a grandparent with Parkinson's, I had an aunt with Parkinson's. But it's such a common condition simply by chance. People are going to have one or two family members. It's not unusual at all. Now, finding families where there is a high rate of Parkinson's, suggesting an inherited pattern, does occur, but it's rare. What we've learned in the last 10 or 15 years is that there are, in fact, risk factors for Parkinson's, and this has come mostly through GWAS work. So this is a so-called Manhattan plot from one of the latest iterations of the GWAS. This is constantly changing. But as you can see, we're up to 37,000 cases of Parkinson's and 1.4 million controls in this, and I think it's even larger now. And so there are many loci that turn out to be linked. This particular one shows 90 independent genome-wide significant signals, but the risk conveyed by most of these individual loci is small. So each individual loci does not have a strong effect on the occurrence of Parkinson's. The signals that really stand out are a couple, and probably the most prominent is this protein, which gene abbreviation is SNCA, or synuclein, alpha-synuclein. And this is clearly one of the genetic contributors to Parkinson's. So genetic variation in the region of alpha-synuclein. Most of what you're seeing here is variation in the promoter region, which affects its expression. Also structural variation in synuclein, so mutations in this protein, although quite rare, do cause a highly penetrant form of autosomal dominant Parkinson's. So the bottom line is many loci are linked, but most of them convey only a small degree of risk. Synuclein is probably one of the higher risk factors, but even there, it increases risk by 2 to 3-fold. It certainly doesn't control appearance of the disease, but a very important gene in the biology of Parkinson's. Synuclein is an interesting story because it was originally discovered in a kindred, the so-called Contorsi kindred, which is in southern Italy and Greece. This wheel in the center shows 11 generations of the Contorsi family. These are have a family with autosomal dominant form of Parkinson's disease. It's also associated with dementia. It's early onset. It's quite an aggressive disease in this family. And this family turned out to have a gene mutation in alpha-synuclein. It's now been found in several other families around the world, also gene multiplications, where the normal synuclein gene is there, but an extra copy is present, so overexpression. Even when this was found, and I remember when this was discovered, this was described in the late 90s, there's a lot of excitement. We all thought, oh, this is the answer to Parkinson's disease. And we went and tested a lot of patients in our clinic looking for synuclein mutations. It's extremely rare. We found none in our clinic at Penn at that time. And then, it went from front burner to back burner when people realized how rare it was. And then Marie Spillantini and others discovered that antibodies to synuclein light up the Lewy body. So if you have a mutation in synuclein, you may have an autosomal dominant form. But even if you don't have a mutation, if you develop Parkinson's, synuclein pathology is going to be present. And the Lewy bodies that appear, the principal component of those Lewy bodies is alpha-synuclein. So I think of this as really a cornerstone of Parkinson biology. And one of the things that evolved from there was the idea that synuclein actually has prion-like properties. This is some work, Laura Volpicelli-Dally from our group gave me this nice figure. And she was one of the people instrumental in showing the ability of synuclein to seed and spread, so that a small aggregate of synuclein can, in culture and in the brain, entrain endogenous synuclein, form fibrils, and these fibrils can propagate from one neuron to another and can spread across the brain. So synuclein has a prion-like aspect when it's misfolded, can lead to spreading disease. And this is a very important idea in the field. And that's been paralleled by work on the spread of pathology in Parkinson's. So I started by emphasizing how important the degeneration of the dopaminergic system is, but recent work has really suggested that that's only a piece of the puzzle, that there's actually a pathologic progression of the disease that matches the clinical progression that we see. So in early stages of Parkinson's, this is work done primarily by the Brocks, you find the pathology in the brainstem and lower midbrain. This may well account for things like the REM behavior disorder and autonomic dysfunction we see prior to motor onset. The mid stage of the disease is when the midbrain is involved and the dopaminergic system degenerates, and that's really associated with classical disease. And then late in the game, you see Lewy pathology spreading across the brain and into the cerebral cortex, and may be associated with a cognitive aspect. So just as on a cellular level, you can see propagation of synuclein from one cell to another, even in culture. Even in all right. So certainly there's a parallel spreading process in the human brain.
I am trying to find out if there's a place that I can.
You may want to figure out who that is and mute it. There you go. All right. So both a microscopic spreading process and pathologically, there appears to be a progression of this disease across the brain. So that's been very important in the field, but it leads to some questions. So you can demonstrate propagation of synuclein in cultured neurons and even in rodents. There's seeding of pathology. But how does this aggregation of synuclein lead to neural injury and degeneration? And is there a component of immune response here? Does alpha-synuclein have a role in actually triggering an immune response? Is that a contributor to what goes on in PD neurodegeneration? I think I'm going to show you a pretty compelling argument that, in fact, the immune response to synuclein is a major part of the destructive process here that's triggered by abnormal forms of synuclein. Before I plow into that, let me talk a little bit about what innate and adaptive immunity are. These are the two major arms of the immune system. Innate immunity is the kind of immune response that's mediated primarily by cells like macrophages, microglia, monocytes, dendritic cells. It's the earliest response to injury or immunologic trigger. And these kinds of responses can be either cytotoxic or protective. So particularly microglia and monocytes have both cytotoxic, destructive abilities, but they also have neuroprotective and neurorestorative abilities. The key thing which distinguishes innate immunity from adaptive immunity, however, is innate immunity is relatively nonspecific. It's triggered by events in the local environment, and it doesn't have a long-term memory of prior exposure to antigen. The memory is really in the adaptive immune system. This is mediated primarily by T and B cells, and they do establish long-term memory of antigenic stimulation. This, of course, is the basis of vaccines as therapies for disease, which is very much front of mind for all of these days. And these interact. The innate adaptive immune system modulates the activity of innate immunity. And in fact, in most settings, to get a full-blown activation of the innate system, you need to engage the adaptive component as well. So what about evidence for immune involvement in human Parkinson's disease? Well, it turns out that we've known for a long time that there is immune activation in human Parkinson's disease. Some of the earliest work, probably by the McGears. This is from a review they published in 2008, but they were working on it for years before that, and showed that there was very prominent microglial activation in postmortem human brain. So this is a figure from their 2008 paper showing CD11B reactive microglia here. So these are activated microglia. in postmortem brain from Parkinson's. You can stain them with HLA-DR. So HLA-DR is a component of the major histocompatibility complex, or MHC. And these glia light up with this MHC or HLA stain. It's a classic sign of activation. And in fact, there were leukocytes found in the brain, and they have one here next to a large melanized neuron. So there are leukocytes in the brain, there are active microglia, and this has been really known for a substantial period of time. Also, you can find IgG in the brain, so this is a stain for IgG. Deposits along the Lewy body. The Lewy body is primarily made of synuclein, but IgG is one component of it. And then you can find CD4 and CD8 T cells in human brain. And this is some classic work from Brochard showing both CD8 and CD4 T cells in the human brain. They quantify it down here on the bottom right. And there really is a remarkable increase in all of this. I think what changed, though, is the thinking that for many years that this was a consequence of the disease. In other words, you had a neurodegenerative state, neurons were dying, and so of course there was an immune reaction in response to this. And what's happened in the last 8 or 10 years is we've turned that around and realized that actually the immune response is the disease. It is a driver of the disease rather than simply a consequence of it. And I think that's an important direction the field is moving. One of the things that led us to this is studies of genetics. So I showed that Manhattan plot before, which was very busy, but this is one that pulls out one particular gene in a group of Parkinson's patients, work done by Haida Payami, who's now here at UAB as well. Here's the synuclein linkage, as we talked about earlier, but a gene that is linked to Parkinson's disease is HLA-DR. Now, HLA-DR is linked to other autoimmune diseases. It's commonly known to be linked to a variety of autoimmune conditions, and the linkage here in a GWAS study with HLA-DR really puts Parkinson's in a class of other disorders where there is a genetic connection between risk of disease and variation in a key immune regulatory protein. There were also evidence for inflammatory markers in blood. So these are, this is a nice meta-analysis. There have been many studies of this over the years, but here they've pulled it together in a meta-analysis to look at various cytokines and chemokines in Parkinson's disease, and a lot of them are elevated. IL-6 is probably one of the ones with the most prominent signal, but also tumor necrosis factor, IL-1 beta, IL-2, IL-10, RANTES, C-reactive protein. So there is a inflammatory signature in the blood in Parkinson's disease. We've gone a little further with this, and this is a recent study that we published last December, looking at inflammation in a cohort of patients with Parkinson's disease. So these are patients we recruited at UAB. These are all newly diagnosed patients. So one of the tricky aspects of Parkinson's is because we're able to treat it, many of these patients are on medications which can themselves affect function of immune cells. So this was a group of patients recruited really at the time of first diagnosis before they were on any treatment for Parkinson's. And what we did was we collected blood samples from these patients at the time of diagnosis. This is a relatively smaller pilot study, but we had 10 males and females, 10 healthy control males, 11 PD, 6 female healthy controls, and 7 PD females. We collected peripheral blood mononuclear cells, and then we isolated blood monocytes when the blood was still fresh, so directly from fresh samples using kind of a typical magnetic bead technology. But this is a negative selection process where we take the cells from blood, we use antibodies that remove everything except the monocytes and leave the monocytes untouched. We can then do a flow cytometric analysis and then also RNA-seq to look at activation states of these blood monocytes. And I'll show you the data in a second, but the bottom line here is, first, the surprising result was there was a very striking effect of sex on monocyte gene expression. So the inflammatory signature you see in men is quite different from what you see in women, which I think is important and ties back to some observations in the field. First of all, that it's more common in men, and that there seem to be some phenotypic differences between men and women in terms of the effects of Parkinson's disease. In females, you see a very clear pattern of inflammatory activation of monocytes, and gene sets that are associated with interferon gamma are enriched. In males, it's a little harder in this set to pull out a signal. I suspect there's more than one subgroup there, and we simply don't have a large enough sample size to see the signal in the males very clearly. Just to show you the data that underlies this, these are some volcano plots of female PD versus healthy control on the left and male PD versus healthy control on the right. You can see in the females, there's a very large difference in the gene expression patterns of females with PDs versus healthy controls. The males, it's murkier, and it's not that there's no signal there. When you really look at individual cases, as I said, I think there are several different patterns, but in the aggregate, it's hard to pull the signal out. We also used a gene set enrichment analysis approach here, where rather than just looking at individual genes, we asked about patterns of gene expression. So for this, we use a set of reference states from the published literature where they'd taken monocytes from blood and activated them using a variety of different triggers with interferon gamma, with LPS, with other triggers for immune activation. We're able to really pull out the gene programs that underlie activation and define the activation states of the monocytes. And several of the modules here, so-called modules 7 and 8, were very much enriched in females. These are modules that are activated by interferon gamma. There's a whole, it's not one gene, it's a whole set, it's a gene program, which is what you'd expect. These are driven by particular transcription factors. But in the females, you see a clear pattern of activation of these two interferon gamma related modules. If you look at those modules in males, again, it's much murkier. Again, I think we just simply need larger sample size to pull it out because we have heterogeneity in the male population. But I think this is important, and this is important to the work we're doing now, because we've realized that we need to actually study males and females separately and have sample sizes that are large enough to actually ask that question. What you often find in Parkinson's studies is many of the research study participants are males. That partly is because of the male prevalence of the disease. And then what you get for controls is you get more females than males, because you tend to get the spouses and care partners of the males come in and participate as controls. So you wind up with groups that are very mismatched with respect to sex, tend to be male Parkinsons and female controls. And actually, one of our challenges has been trying to work against that and come up with balanced groups where you can actually ask questions about the sexes separately. I think this is a study that has really clued us into the importance of that. So what about adaptive immunity in Parkinson's? Can you also see evidence for adaptive immunity in the blood of Parkinson's patients? And this is a paper, work done by David Sulzer now about 4 years ago, that I think has really turned the field on its head. So what he did with his colleagues is he isolated T cells from the blood of patients with Parkinson's disease and asked, can we see T cells that have a memory of alpha-synuclein? And to do this, they used a library of peptides, you can see them across the bottom right here, and screened the T cells for reactivity against these different peptides. And these form a tiling of the alpha-synuclein protein. And what you can see is that measured by response with interferon gamma, colony-forming groups, or IL-5, any of these, there's a region of the synuclein protein here that is a strong antigen, and there are T cells in the blood of patients with Parkinson's that have a memory of this region of synuclein. And in fact, this is the site of phosphorylation of synuclein. This is near the so-called S129, which is a site that's often abnormally phosphorylated in Parkinson's disease. So this was pretty striking. I didn't think a lot of people didn't think you would be able to find a T cell memory in the blood in patients with Parkinson's disease. And in fact, there's a deeper story here when you look at HLA linkage, that the genetics of the patient determine the immune response that they have to alpha-synuclein, linking back to the original discovery of the role of HLA as a risk factor for Parkinson's. So this was important. And then we collaborated with David Sulzer and Cecilia and the others to extend this in another paper looking at a larger sample. And here they tried to look at the question of cross-sectionally across the duration of Parkinson's, when do you see this T cell immune response? And the graph on the right here, it starts with years since diagnosis, with 0 over here on the left, and going out to 30 years after diagnosis. And this is the measure of the T cell responsiveness to alpha-synuclein. The T cell response is highest at the time of diagnosis. And after about 10 years, it fades away. Even though the disease continues, the patients continue to have Parkinson's beyond 10 years. But the highest activity of the T cell response is early in the game. And then this graph on the bottom left is interesting in the sense that this is a fortuitous occurrence. A patient who was an immunologist and actually collected his own peripheral blood mononuclear cells for several years before developing Parkinson's. Turned out to actually have T cell reactivity to alpha-synuclein prior to the onset of the disease. Now, this is a N of 1 case. We don't know if that's going to replicate. Not too many people store their PBMCs in the freezer for no reason, not knowing they'll get Parkinson's in the future. But obviously, this is perhaps a clue, and now we're going back to high-risk populations of Parkinson's and trying to look at this further. But this really emphasizes the T cell response and points to the idea that it's early in the game when you see the most vigorous T cell response in Parkinson's disease. I wanted to talk just a little bit about the gut and gut dysbiosis. There's been a lot said about the role of the microbiome and the gut in Parkinson's disease. What is the trigger for these immune responses? And there's been a lot of interest in the question of whether microbial organisms in the microbiome could be part of what fuels the immune response to Parkinson's disease. This is a study we did with Haida Payami, doing a microbiome-wide association study. So this is an MWAS, very similar to a genome-wide association study. It's a pretty large sample, 840 subjects. About 500 of those are from UAB and others are from other sites. Parkinson's in control. And using this large cohort, she was actually able to pull out 15 genera of bacteria in the intestinal microbiome that were associated with PD. And these fall into 3 clusters. Cluster 1 are genes that are really opportunistic pathogens. In many circumstances, they cause human infection, human disease. They're not often seen in normal microbiota, and these are increased in Parkinson's. There's another group that are small chain fatty acid producing bacteria, which are reduced in PD. Others have seen this as well. And there's a cluster 3, which are carbohydrate metabolizers. But if you look at these linkage maps, these are correlation analysis. The genes that are the microbial pathogens here are really this cluster up here in the top. So there's a group of related genes that are upregulated in Parkinson's disease. And so, is this a contributor? Does this create an inflammatory state? Is this a contributor to the eventual immune activation that occurs in Parkinson's disease? I think we don't know, but it's a really important question. We're actually doing studies now using gnotobiotic mice to try and take some of these organisms that we've identified in human microbiota and repopulate animal models with them to ask whether it modifies the response to alpha-synuclein. So don't have the data on that yet, but we're working on it. What about studies in living patients? Is it possible to see inflammation in human Parkinson's disease? One of the major tools for this is, of course, brain imaging. There have been a number of studies done using different ligands. Most of these bind to the so-called TSPO protein, which is a protein that's abundant in microglia and macrophages. PK11195 was an early tracer. More recently, DPA714 and DPA713 have been used. All of these show an increase in uptake in early Parkinson's disease patients, and I think are an important technique. So one of the things we're doing, I direct the Alabama Udall Center, and so our Udall Center is really directed at asking the question of, is there evidence for inflammation in early Parkinson's, and does the presence of inflammation predict the outcome of these patients? And so the clinical core of this, which is run by my colleague, Dr. Tlain Yakubian, is recruiting and assessing a total of 120 patients with PD and healthy control. And you can see the accrual diagram here on the right. This goes out to May. Our goal is 60 in each arm, and we're actually nearly there. As you might predict, the most difficult subjects to recruit have been the female PD and the male healthy control. So we're missing a few of those, but we should wrap this up in the next couple of months. And what we're doing with these subjects is collecting a broad range of biospecimens. They all get brain imaging with DPA 714 to assess central inflammation. They get lumbar puncture, they get blood collection. These data are banked both locally and they're also deposited in BioCent, which is the NIH biobank. And so we hope to use this to ask the question of whether there is inflammation in this early cohort. And then in terms of measuring progression in this group, we're actually using cognitive measures as our primary outcome. These patients get detailed neuropsychometric assessment that's done annually, and that's actually a more sensitive way to follow them forward. So we should complete the baseline group and be able to start talking about the baseline characteristics of the Alabama U cohort by later this fall. What about treating Parkinson's disease with immunotherapy? Is this the future of this? Is of course where we're hoping this goes, and we're hoping that you can lay the groundwork through work like what we're doing in the Udall Center. But the big questions are, of course, can immune modulation modify the course of PD? What are the targets for immune modulating therapy? And when in the course of disease is immune modulation effective? So one observational piece of data that's important to know about is this study, which looked at anti-tumor necrosis factor therapy in the incidence of Parkinson's among patients with inflammatory bowel disease. So anti-TNF therapy is a major form of therapy for inflammatory bowel disease. It's quite effective in treating IBD. And so many patients with IBD are treated with anti-TNF therapy. What these investigators did is use the Medicare database to look at individuals who had IBD and were treated with anti-TNF agents and ask about the incidence of Parkinson's disease in this group. And then they matched, so they had 144,000 individuals. They matched it with 700,000 controls. And among all of this, they found 1,796 that eventually developed Parkinson's disease. So what they discovered is, first of all, IBD increases the risk of PD. That's not too surprising. What you're really saying there is one immune disease increases the risk of another, and that's a common observation. But the anti-TNF treatment of IBD was remarkably effective in reducing the risk of Parkinson's, a 10-fold reduction in the risk of Parkinson's. Now, that's a really remarkable effect. If that was reproducible in a prospective sense and we could reduce the risk of Parkinson's by 10-fold, that would be really a major accomplishment. Now, of course, this is retrospective. It's not randomized. There could be other factors. This could be an association and not a true cause and effect link, but it's a very intriguing observation that makes you think that treatments like inhibiting tumor necrosis factor could be important in prevention of Parkinson's disease. So how do you study that? We've studied experimentally using a number of different animal models. One of the major models we use is this alpha-synuclein model, where we take a rat brain and we overexpress alpha-synuclein using an adeno-associated virus. And the nice thing about this is it's a fairly easy technique. It's very reproducible. You can induce overexpression of synuclein in pretty much any mouse. So you can use a variety of genetically modified mouse strains. And it gives you a really reliable phenotype. There's cytokine production at 2 or 4 weeks. There's IgG deposition. There's microglial activation. You see lymphocyte infiltration. And if you wait about 6 months, you'll see about 30% loss of Th neurons in this rodent model. So we've used this to ask some of these questions about, well, what might the targets for immune modulation be? And so some of the characteristics of this model are shown here. Alpha-synuclein overexpression gives you, as I said, cytokine production, IgG, microglial activation. I'm an anatomist, and I always studied this by immunocytochemistry, but my lab has convinced me that flow cytometry is the way to study immunology in the brain. And so we do that quite a lot as well. These are some flow plots showing our control and our synuclein virus. Gives you a big increase in lymphocytes as well as monocytes and macrophages in the brain. You can see down below here, we get a substantial increase in activated microglia. We get infiltrating monocytes. So there are monocytes from the blood which enter the brain, and we've done this using a variety of different strategies, both labeling for particular epitopes and also genetically tagging them. And there's infiltration of T cells into the brain in response to alpha-synuclein overexpression. We've looked at some strategies that prevent or reduce alpha-synuclein toxicity in model systems. So if you delete MHC2, you can overexpress the synuclein in the brain, and it's not toxic any longer. It doesn't kill the dopamine neurons if you delete this major histocompatibility component, which is important for activation of innate and adaptive immunity. If you silence C2TA, which is an upstream transcription factor, that's also very effective in preventing alpha-synuclein toxicity. If you delete CCR2, which is the one of the triggers for monocyte to enter from the blood into the brain, that's another effective way to reduce toxicity of synuclein. You can inhibit or delete LRRK2, another one of the proteins linked to Parkinson's, and we think that's because LRRK2 is actually highly expressed in monocytes and has a key role in immune regulation. You can block JAK signaling, which my colleague Dr. Benvenisti is very interested in. This is a target of treatment for other immunologic diseases as well. We've also looked at microRNAs, deletion of a particular microRNA 155, which is involved in monocyte activation. So all of this is published work and shows that there are a variety of different ways that you can block immune responses to synuclein and reduce neural injury. I wanted to show some of the more recent work where we've pushed into the adaptive area and asked about T-cell strategies. Can we prevent the toxicity of synuclein by modifying T-cell responses? As I showed earlier, T-cell responses are present quite early in human PD. There are a lot of therapies based on T-cell responses, widely used in MS, other autoimmune disorders, so there are drugs already approved and on the market to modify T-cell biology that might be adapted for Parkinson's. And we thought that probably CD4 T cells would be required. This would be the most likely suspect, and set out to look at that. So this is work done largely by Greg Williams, who was a student in the lab with Ashley Harms. He's now gone on to a postdoc in San Diego. So he started out by looking at using this viral model, overexpressing synuclein, and looking for T cells using staining for CD3. And what you see is there's quite a remarkable increase in the number of CD3 positive cells in the brain in response to synuclein. You can see them up here in the top right, where the green is phosphosynuclein and the red is CD3. You can also quantify them down here in the bottom right. You can subtype those by looking at both CD4 or CD8 staining. Probably easiest to see in these bottom panels here. These are the alpha-synuclein treated animals blown up here, and shows you all of these red cells. Here are CD4, these are CD8. So both CD4 and CD8 T cells are entering the brain in response to alpha-synuclein overexpression. You can quantify that, and you can see there's a very dramatic increase in both categories of cells. What are these cells doing? Well, he did some intracellular staining and flow cytometric analysis of these cells from the brain. Turns out many of them are interferon gamma producing cells, so they're interferon gamma positive. Again, linking back to our findings in human PD, where we found that one of the signatures of monocyte activation is also responsiveness to interferon gamma. And this is usually a pro-inflammatory kind of destructive cytotoxic cytokine. What happens if you delete T cells from the brain? So there are a variety of different ways of doing this. And so Greg did this genetically. He used here TCR beta knockout animals. So these TCRB knockout animals have no T cells. And if you inject AAV2 synuclein here, he's using MHC2 here as a marker of monocyte activation. That goes away. And if you do this quantitatively with flow cytometry, you can see that eliminating the T cells prevents the monocyte reaction to alpha-synuclein. So of course there are no T cells in the brain, these animals don't have them, but the absence of the T cells also prevents the activation of the innate immune response here as well. So that's an effective way to block inflammation. What about is it CD4 or CD8? And so he went on to use animals with selective deletion of CD4 and CD8. And here showing you really the key outcome measure of loss of dopaminergic neurons at 6 months. So over here on the right, you can see these are wild type animals. These are the controls. These are the ones treated with synuclein. They have about a 30% loss. If you knock out CD4, you completely prevent the toxicity. The synuclein is still there. It's still very abundant. It's still aggregated. There's high molecular weight synuclein, but it doesn't destroy the neurons if there's no CD4 cells. CD8 did not block the toxicity. So at least in this model system, CD4 seems to be the key subset, in CD8 deletion does not prevent the toxicity of alpha-synuclein. Can you do this pharmacologically? So there are a number of different ways of preventing T cells from entering the brain. Many of these are developed as therapies for MS. Here he's used a drug called FTY-720, also known as fingolimod. Perhaps some people out there have treated patients with fingolimod. This is commonly used in MS. And what it does is, of course, marginalizes T cells from the circulation to the lymph nodes, and so there are fewer to enter the brain. And here you can again see with this label of MHC2, these are the vehicle animals here. This is the AV synuclein. So here's the control. Here's the the fingolimod treated animals. So there's a big reduction in inflammation in the brain when you treat with fingolimod. So this is another pharmacologic strategy for reducing the inflammatory response to alpha-synuclein. So some key points to take away from all this. One is there's a lot of evidence for immune activation in human PD. There's clearly microglial activation. There's monocyte infiltration. There's brain T cell infiltration. There are increased blood cytokines, monocyte activation, circulating alpha-synuclein reactive T cells. And there is gut dysbiosis, and how that's connected exactly to this is a kind of a chicken and the egg question. Is the gut dysbiosis the cause of the brain immune response, or is the Parkinson's disease and the consequent changes in gut motility the cause of the gut dysbiosis? That's something we need to untangle experimentally, but the observation in humans is very clear that there is abnormal microbiome in human Parkinson's. In animal models, you can replicate a lot of these features of inflammation. And inhibition of any of several different immune signaling pathways can prevent alpha-synuclein-induced inflammation and neurodegeneration. So there's not just one target, there are a number of different ways of modifying the immune response. CD4 cells do seem to have a critical role, and this may be an important target. As I said, there are drugs already approved and marketed which have an effect on T cells, and ways of thinking about treating Parkinson's with some of those would be really interesting. So another way to put this together and think about it, the way I think about inflammation and its role in Parkinson's disease, I think of it as the driving force of progression. I do get the question of, do I think the immune system causes Parkinson's? I don't really think so. I think the causes of Parkinson's are the things many people in the field have studied for a long time. Alpha-synuclein has a critical role, and the misfolding of alpha-synuclein is very important. Oxidative stress is the oldest story in Parkinson's. It's the one that really helps us to understand best why the dopamine system is so vulnerable as opposed to other areas of the brain, and probably has a role, probably promotes misfolding of synuclein or impairs its clearance. And then environmental exposures are clearly linked to Parkinson's. There's pesticide exposure and other kinds of environmental toxins clearly have a role, and these are proximate triggers. But once somebody has Parkinson's, or once the disease process has been initiated, the misfolded modified alpha-synuclein becomes a key feature. That drives MHC2 mediated antigen presentation. That drives downstream cytokine and chemokine induction, ultimate recruitment of a T cell response, microRNA modulation, and infiltration of peripheral monocytes. And that's what ultimately leads to the destructive process. So for me, when I see a patient with Parkinson's in the clinic, it may be true that their disorder is caused by alpha-synuclein, oxidative stress, environmental exposures, other upstream features, but most likely that all happened a decade before I saw them. For the patients who's sitting in front of me, what is the process that's making them worse from one year to the next? And I think there's a good argument to be made that the inflammatory process is actually what's driving the progression from one year to the next, and is probably the best approach to therapy in people with established Parkinson's disease. I do want to thank our team. This is the Alabama Udall Center team, which is quite a large group, as you can see. We also enjoy support from the American Parkinson's Disease Association and the Parkinson Association of Alabama. So a lot of people are involved, but you know, a lot of the work I showed you was from my own lab team, and of course, the way my lab meets now looks like this. It doesn't look like a meeting around a table anymore, but this is some of the lab group. Ashley Harms, who's now my long-term collaborator, has been really key in this, and a lot of the T-cell work here was done by Greg Williams and his graduate school colleague Aubrey Schoenhoff, both of whom are in San Diego doing postdocs now. So I can stop there, and I'll be happy to take some questions.
Okay, I'm going to ask the first one. Don't you want to just get that Contour-C cohort and put them all on some sort of anti-inflammatory? Or I don't know, maybe they're already being treated. Maybe somebody's already.
Well, I doubt it. So, you know, anti-inflammatory treatment in Parkinson's is, you know, it's a risk-benefit trade-off, and you have to decide where to draw the line. There is some evidence that things like non-steroidal anti-inflammatory medications may reduce the risk of PD a little bit, maybe up to 15%, but they have negative effects on kidney function and gastric bleeding and other kinds of adverse effects. So I don't recommend that. I do wonder about T-cell based therapies. So drugs like Tisabri, which is an alpha integrin blocker and prevents T-cell entry into the brain. It's an approved therapy for MS, and one wonders if that would be an approach in Parkinson's disease, or you know, other kinds of anti-inflammatory drugs. There's interest in the inflammasome, NLRP3 inhibitors. There are a variety of ways of tackling this. One of the keys of doing human work, though, is that you have to have biomarkers of your response. You know, this is we've tested so many therapies in Parkinson's, and most of them have failed. And one of the reasons they failed is we had no way of knowing if they were doing what we thought they were doing. You know, if you try an antioxidant in Parkinson's and it doesn't work, like vitamin E has been tested, it doesn't work. Did that not work because it did not reduce oxidative stress in the brain? Or did it not work because that just doesn't work? We don't know because we had no way of measuring brain oxidative stress. So if you're going to inhibit the immune response, I do think it's really important to have a handle on how do we measure that immune response, and how can we see that our drug's reducing it before we invest many millions of dollars to see if it alters the course.
Hey, David, I have a question. So many, I mean, it seems like there's a trend of neurologic disorders, right? Or degenerative disorders. I mean, the inflammation somehow evolved.
Mm-hmm.
You think, so what makes the Parkinson's disease unique? Is the Lewy body that starts the whole thing? Or why the consequences are not more general in other disorders? You know
well, I think the immune response is involved in many neurodegenerative diseases. There's good evidence in Alzheimer's, for example, that the immune response is important. Also ALS, quite a lot of work's been done in ALS and showing the role of the immune response. I think the specificity, the immune system's activated by particular antigens. And my feeling is that, you know, synuclein is probably one of the antigens that drives the response in Parkinson's. So the reason you see the pattern of pathology that you do is because the underlying driver of this is abnormal and misfolded synuclein. And so where you find the synuclein is where you're going to find the immune response. And there is a very strong correlation between the location of abnormal synuclein and the location of activated microglia in the brain. There's also abnormal synuclein in the gut, in the walls of the gut. You can find it in the heart. And probably some people even know one of the big trends in Parkinson's these days is diagnosing Parkinson's by skin biopsy. So if you biopsy the skin in a Parkinson's patient, you can actually see abnormal synuclein in the small unmyelinated nerve fibers. So Parkinson's is a whole body disease, and the antigen is in a number of places. And one of the important questions for us is where does it start? Where does this all begin? What is the launching pad for all of this? But I think the specificity is driven by, you know, what is the nature of the proteins the immune system is responding to.
David, there's a question for you in the chat.
Mm-hmm.
Do you have access to it?
Yep. So a question about genetics in Parkinson's disease, and this is one I get a lot from my patients. I would say almost every patient, when I diagnose them, first thing they say is it genetic and my children are going to get this? And generally speaking, the answer in the clinic is no, your children are not at more risk than anyone else. The genetics of Parkinson's, for most people, the inherited risk from one generation to the next is pretty small. Now that GWAS does show you there are 90 loci linked to Parkinson's disease, and each of those has a small increase in risk. And there probably is a polygenic component here where those loci together may account for a significant piece of the risk, but that's not something you're going to see in a family. There are families which have high risk of Parkinson's. The Contorsi kindred's one of them. LRRK2 is one of the commoner mutations, and people with LRRK2 mutation in the family do have certainly an increased risk and often an autosomal dominant pattern of inheritance. Also GBA mutations, another group of people with a higher risk of Parkinson's. So you can identify some of these genetic forms, but you know, for 95% of the patients, it's not going to be a strongly familial disorder. And what I do practically in the clinic is someone says, my grandfather had Parkinson's, I have Parkinson's, are all my children going to get Parkinson's? I say, no, that your odds are no worse than anyone else. Now, if they say, I have 8 relatives with Parkinson's, that's a different story. And that's where we pursue genetic testing and really try to understand that. Also very young onset Parkinson's. So Parkinson's that begins before the age of 40, which is a rare situation, has a much higher chance of being driven by a gene, often one of the autosomal recessive genes. So I don't know that testing at birth is going to get us anywhere unless you're from a family that's known to have a causative gene, and those are quite rare. Is there another question in here? I think you're muted, Eleni.
Oh, there you go. I was taught, and maybe this is no longer the thinking, that Parkinson's is an ascending disorder. And I'm wondering if there's anything about the way the immune response is generated, or the way it works, that would explain that phenomenon, or whether that's no longer the current thinking.
I don't know about an ascending disorder. I think we've recognized that there are prodromal features that appear before motor Parkinson's. And those prodromal features, so REM behavior disorder is one of the earliest and most distinctive ones. But constipation is perhaps the earliest feature that you can identify in Parkinson's disease. And there's, I think the most dramatic illustration of that is from the Honolulu Heart Study. So the Honolulu Heart Study was a study of men that was launched in the 60s in Honolulu to ask about cardiovascular risk factors. But they did gather a lot of data about them and have gone back and looked at development of Parkinson's in these patients. So the strongest risk factor for Parkinson's 30 years later in this group was constipation. If you had less than 1 bowel movement a day, you had a 12-fold increase in the risk of Parkinson's 30 years later. So, that's a very early feature, and makes you think that some of this pathology in the gut may develop very early on. So I don't know about ascending. It's a whole body disease, and there's definitely a prodrome, and a lot of work is going into that. Fox Foundation is now, they've done some large studies of early PD, but they're trying to back the clock up and get people who are at risk by going to families with a strong family history, by going to people with REM behavior disorder, by going to people with constipation and anosmia. And can you, out of a population, find some people at high risk?
Well, speaking of high risk, there's a question in the chat.
Mm-hmm. Viral infection development of Parkinson's. Well, so, the oldest story around this is the influenza epidemic of 1918, right? So the pandemic flu, the so-called Spanish flu of 1918, did lead to a very large number of patients developing Parkinson's disease. Many of them had encephalitis as part of the flu, and later Parkinsonism was found to be a major component of that. That's actually been studied in animals. Richard Smaney and a few others have studied it. Obviously, it's very difficult to work with pandemic flu. It's a very dangerous organism, and not too many people work with it, but a few people have looked at it, and it does seem to have a tropism for dopaminergic neurons and produce destruction of the dopaminergic system ultimately. Is that immune mediated? I don't think we really know. We don't have very many brains around from 1918. We don't really have, it's never really been looked at. I would wonder if there's an immunologic aspect to the Parkinson's caused by pandemic flu. Of course, everyone's wondering about coronavirus and what that will do. We don't know. It's a little scary that it causes loss of the sense of smell, which is another characteristic feature of Parkinson's. And certainly we know Parkinson patients do very poorly when they get the coronavirus, but in terms of what that will be like in 10 or 20 years, no one knows.
Well, is there any other questions? Well, if not, thank you, David, for this very exciting and stimulating talk.
All right. Well, I enjoyed talking, and I guess I'm going to rejoin in an hour or two here, right?
Okay.
That sounds great.
All right.
Thank you so much.
All right. I'll see you guys a bit later.